Hello, everyone. Here we are in chapter 6, third week, and we're talking about microbial nutrition and growth. This chapter has a few very neat things that had to deal with metabolism. Okay. We're going to start with oxygen requirements. We have what's called obligate aerobes and obligate anaerobes. So obligate aerobes are organisms such as you and me that must be in an oxygen atmosphere, otherwise we're not going to live. So that means that we're running glycolysis, Krebs cycle, and the electron transport chain, okay? And subsequently, we need oxygen as the final electron acceptor in order to be able to breathe. So we need oxygen. That's an obligate aerobe. And the other version is obligate anaerobe. Okay, so there's two flavors to this. As an obligate anaerobe, you're either not running the oxygen portion of catabolism, which means that you're running glycolysis, and you're running the you're running glycolysis, but you are not running the Krebs cycle or the electron transport chain. That's one thing it means. The other thing that it means is that you are running glycolysis, Krebs cycle, and electron transport chain, but the final electron acceptor is not oxygen. It's either sulfur or nitrate, okay? In either of those two instances, these organisms cannot live under the presence of oxygen and are consequently called obligate anaerobes. Now, as it turns out, oxygen is a necessary evil. Okay, we need it in order to produce energy, and again, its requirement is the fact that it accepts electrons readily, but it's something that we have to kind of put up with. Okay, we have to defend ourselves against the problems that oxygen brings up, and bacteria has to do the same thing. So one of the things that's important to bring up is the toxic forms of oxygen that both bacteria and we fight against, okay? So if you look on this slide, you can see that we have the so-called singlet, okay? This is an oxygen molecule, see, O2, but it's a little one on there, which means that there's an electron, not an extra electron, it's just part of the electron of the molecule that is high energy, it's got more kinetic energy that it knows what to do with, and it's unhappy. Okay, it's going to stay unhappy until it loses that kinetic energy or carries out some kind of reaction that allows it to lose that kinetic energy. So consequently, you end up with this problem in that the singlet electrons can react indiscriminately. Now, how do bacteria and our cells protect against it? We use this thing called a carotenoid. Okay, so carotenoids. Let me bring up one here. Okay. And what you'll see is this long molecule that has alternating single and double bonds. Single, double, single, double, single, double, okay? That's typical of carotenoids. Why do we care? In this alternating single-double system that is said to be conjugated, okay, because it's single-double, there's p orbitals, both above and below the molecule, that can accept electrons readily, okay? As a consequence, these carotenoids, these molecules, can play hot potato with this high-energy electron until it decides to give up its kinetic energy, and then it calms down, okay? So this is what bacteria and ourselves use in order to protect ourselves against these singlet oxygens. Even worse than singlet oxygens are these superoxide radicals, okay? Superoxide radicals, notice we still have that oxygen molecule, but this time we have a negative on there. That negative means that now we actually have an extra electron. That's actually worse, okay? The extra electron is going to be unhappy until it finds a home. How do we protect ourselves? Well, both bacteria and ourselves use superoxide dismutases. These are types of enzymes that use a metal a core in their active site, zinc, manganese, iron, nickel, or copper, one of those. And what they do is they turn that superoxide, they combine it with protons, and they produce hydrogen peroxide, okay? Now, that seems like a good thing, except that hydrogen peroxide is actually worse. So what happens now? Well, we go to the next level here, the peroxide anion. Notice we still have our oxygen molecule, but instead of having one extra electron, we have two extra electrons. These electrons are very unhappy, okay? The only thing worse than one electron with nothing to do is two electrons with nothing to do. So here it is. There's a peroxide anion, okay? You and I know it as H2O2. And what protects us against it is this enzyme called catalase that we have in our blood, okay? So it's produced in the liver, it goes into blood, and this catalase turns this hydrogen peroxide into water and oxygen, both of these being benign, and now we're okay, okay? So we only use catalase. Bacteria, some bacteria use catalase, some use peroxidase. So the end effect is the same, and that's the fact that we are taking something very dangerous, which is a peroxide ion, and turning into something very benign, such as water and oxygen, or water and NAD in the case of peroxidase. The last one is the so-called hydroxyl radical. The hydroxyl radical has an extra electron, and these guys are really hard to deal with. As a matter of fact, we don't have anything to deal with them, okay? The only thing that I can tell you is that what we try to do is prevent their formation by keeping the concentrations of hydrogen peroxide as low as possible, and therefore the incidence of this hydroxyl radical as low as possible. Now, if it does happen to manifest itself in our tissues, antioxidants such as vitamin C and vitamin E can protect us to some extent against these radicals. So why are we covering this? Because, well, number one, it's important. It explains to you why people keep on talking about antioxidants. Antioxidants are necessary because they continue to protect our body, okay? They protect it against oxygen. But the other thing that's interesting is that if you have an organism that's an obligate anaerobe, in other words, it evolved in an environment where it was not accustomed to being in the presence of oxygen, it might not have these defense mechanisms such as carotenoids and dismutases and catalases and peroxidases. These organisms are usually Clostridia, okay? So C. difficile, I'm sure maybe you've heard of it. C. perfringens, maybe you've heard of it. And these guys cannot survive very well in the presence of oxygen. So if you have oxygen, these toxic forms have a tendency to develop themselves around them and kill this Clostridia. And thank God, because Clostridia, these obligate anaerobes, are very bad pathogens. So different traits that you're going to find regarding oxygen requirements is aerobes, okay, that require oxygen, anaerobes that do not require oxygen, facultative anaerobes that can either take it or leave it. An example is E. coli. You can either use oxygen and use glycolysis, Krebs cycle, and electron transport chain, or you cannot use oxygen, only use glycolysis and still be equally happy. So E. coli can grow under both conditions, better under aerobic conditions because it produces more ATP, but it does not have to be aerobic conditions. Aerotolerant anaerobes are anaerobes, organisms that do not require oxygen at all. However, they have some protection against oxygen. Maybe they produce carotenoids. Maybe they're catalase positive. Either way, they can protect themselves against certain oxygen species, and consequently, we call them aerotolerant anaerobes. And then microaerophiles are organisms that require small amounts of oxygen. They have a slow metabolism and require oxygen, but a very slow rate. So what would this look like? This is what it would look like. Imagine that this is a liquid media, okay? This is what the obligate aerobe would look like. The majority of the growth would be towards the end where the oxygen is at. And when there's no oxygen at the bottom, there would not be any growth. With the obligate anaerobe, the opposite would be true. It would grow where there's an absence of oxygen at the bottom, And where oxygen diffuses through the top surface, there would be no growth. Facultative anaerobes like E. coli, again, they prefer to be in oxygen, but if there's no oxygen, that's okay. They'll grow anyways, just not as fast. So you'll see a turbidity that is high at the top and slow and decreases as it goes down, but never goes to zero. And then aerotolerant anaerobes, these are organisms that do not require oxygen, but have some of the defense mechanisms that allow them to live in oxygen, and consequently the turbidity would be uniform throughout the tube. So these are good ways to discern the oxygen requirements of microorganisms, and then therefore find out what they are. Okay, we're moving to other nutrients. We're looking at nitrogen. Okay, nitrogen has a couple big roles in our body. First of all, it's a part of amino acids. So therefore, it's part of the prerequisites to making proteins. Another thing that nitrogen does is that it's part of the nucleic basis. So if you're making protein, if you're making DNA, if you're making messenger RNA, you're going to require nitrogen. Okay, this is true of us, this is true of bacteria, this is true of fungi, this is true of plants, this is true of all life. Okay, bacteria, however, are incredible in the fact that they can carry out what's called nitrogen fixation. So our atmosphere is about 79 to 80% nitrogen and only about 18 to 19% oxygen. So there's a lot of nitrogen, almost 80% nitrogen in our atmosphere. Yet, in our agriculture, sometimes we cannot grow plants in agriculture because there's no nitrogen in the soil. We have a nitrogen poor soil. So what do? You grow a type of plant called a legume. These legumes grow nodules that attract nitrogen fixating bacteria to it. And what these bacteria do is they have the ability to turn this atmospheric oxygen into nitrates that can then be assimilated by the plant and consequently by animals later on. So bacteria are incredible because what they do is they open a resource of nitrogen that is not available to any other organism. Only nitrogen fixing bacteria can do this. So they turn the nitrogen in the atmosphere into useful nitrogen for you and me. Phosphorus, okay, phosphorus is very important. It makes a phosphodiester backbone of nucleotides and mRNA. But you also make ATP, GTP, CTP, and GTP, the nucleotide triphosphates, okay? So you should know by now that ATP, adenosine triphosphate, is the energy currency of the cell, okay? So without phosphorus, you wouldn't have energy. GTP is frequently used as an energy currency in the body as well. So that would be another type of energy that would not be available to our bodies if we can assimilate phosphorus. So again, phosphorus necessary for the building of nucleic acids such as DNA and RNA, and to produce energy such as ATP and GTP. Then we look at the next one, sulfur. Sulfur looks trivial. Sulfur, it gets incorporated into one of the 20 amino acids. The name of the amino acid is called cysteine. You might think that this is trivial, but as it turns out, cysteine makes one of the strongest interactions of all amino acids. They make a covalent link between sulfurs called a disulfide bridge. And because this interaction is so strong, it means that proteins that require to maintain a certain three-dimensional shape are typically rich in cysteine. The more important it is for that molecule not to change shape, the more likely that it's going to be richer in the amino acid cysteine. Okay. Growth factors. We consider growth factors any large molecule that is a necessary precursor to life. This could be vitamins. It could be shuttles such as NAD or FAD. It could be lipid arms that are cofactors and enzymes. So any large molecule that is organic, carbon containing, and it's an essential precursor to life, it's called a growth factor. Okay. pH. So we have two most organisms in our body are accustomed to growing at a certain pH. The flora on our skin, for example, has a tendency to live in a slightly alkaline environment. The flora in our stomach, because of the hydrochloric acid in our stomach, grows at a low pH, because acidic is low pH and alkaline is high pH. So whatever the organism has evolved to adapt itself to, that's the pH in which it's going to grow most efficiently in. So having the right pH is an important precursor to the life of microorganisms. This talks about the creation of biofilms. So you can see we've talked a lot about biofilms. This week in the news, I talk about, or don't talk about, I give you an article about colonoscopes and maybe sigmoidoscopes that are developing biofilms that are being transferred from one patient to another. So effectively what this means is that when these bacteria come together and they settle on a surface to start working synergistically, and then it is very, very difficult to get rid of them. Bleach might not work against them. Alcohol might not work against them. And as you can see, once these cells come together, okay, to start what is called quorum sensing. So quorum sensing means that they release molecules to communicate with each other, find out who else is here and how many of them there are. When they communicate, they start working together to start making this so-called extracellular matrix that protects them against the environment. And then once they know that they're protected against their environment, then their pathology changes and they start misbehaving. So this is very interesting because these bacteria, these prokaryotes, usually behave very nicely until they feel defended by this biofilm. And then once that biofilm is there, then they start showing they start expressing their pathogenic genes. So the problem is that once this matrix is there and these organisms come together, it is very, very hard to get rid of these biofilms. Okay, let's see. All right, let's keep on going. The next interesting trait about organisms, we addressed this in chapter 1 when we said that Dr. Koch went from liquid media to solid media, and this was significant because we could discern the identity of microorganisms based on their macroscopic morphology. Okay, so this is what this is addressing, is macroscopic morphology. In other words, how does the colony of a particular organism grow on solid media? So things to discern is shape, how the overall colony grows. And I would like for you to assimilate this jargon because it's the way that microbiologists and pathologists talk about these organisms. So you can see circular, rhizoid, looks like it's the bacterium is growing in a root shape. Irregular, filamentous, that look like mycelia. Spindle, there's some lactobacillus that grow in a spindle shape. So this is the overall shape of the colony. The margin, we're looking at just the edge of it. So if it's smooth, it's entire. If it's sigmoidal, it's undulate. If it's even higher, more lobate, if it's even more sigmoidal, then it's called lobate. Here's curled, okay? I don't know if I talked to you about curled before, but if you look at Proteus mirabilis, okay? Proteus mirabilis, and we look at an image of it, we come up to a plate that looks like this. See that? So what happens here is that they had a Petri dish, and they inoculated the very center of the Petri dish with this Proteus mirabilis, and the Proteus mirabilis started swarming out until the nutrients were dissipated. Then they swarm out, nutrients would dissipate, they swarm out. And so you end up with this pattern here. And as you can see, it matches very much this curled margin that we're looking at here. Okay, filiform. Okay, looks filamentous in shape. If you turn the petri dish on the side, some colonies are flat, some are raised, some are convex. A lot of this has to do on how fast the cells divide. Okay, at the worst case scenario, they divide very quickly. They end up with a low hill, and then the cells in the middle die, and you end up with this shape called crateriform, meaning in the shape of a crater. Okay, size, Moraxella catarrhalis, for example, is punctiform because the size of the colony is very small. Or you can go to Bacillus megaterum, where the colony is very, very large. The texture, if the colony is smooth or rough, that means that it's different organisms. Is it glistening or dull? K. pneumoniae, for example, has a mucoid look to it and is very shiny. So if you see a mucoid or shiny colony, you can tell that it's K. pneumoniae. Pigmentation, you can have purple bacteria, red bacteria, yellow bacteria, buff bacteria. All those different colors are different types of bacteria. Okay, and then is the cell transparent? Is it translucent? Can we see light through it? Is it opaque? Can we not see any light through it? So all these are traits that allow us to describe macroscopic morphology effectively. Okay, let's go to streak and pore plates. One of the things that you're going to be doing soon here, I hope, is you're going to be able to do what's called a streak plate, which means you have a petri dish and you can take a you can take a loop, and you're going to do 1, 2, 3, and 4 streaks. And as you can see, here's the end result. What we're trying to do is we're trying to decrease the cell density so we can see independent cells at the end. Okay, so this is called the streak plate method of isolation. You streak once, and then with a clean loop, you streak into the first one. With a clean loop, we streak the third one into the second one, and with a clean loop, we streak the fourth one into the third one. And you end up with this isolation that you see here. Let's see. This is the pore plate that I was alluding to in the lab, where you carry out dilutions, and then you inoculate the plate with 0.1 mLs. Here it's showing 1 ml, but typically it's 1/10 of a milliliter. And what that allows you to do is to lower the density of colonies in a petri dish so you can isolate 2 organisms that are typically very difficult to isolate. Okay. Now we're going to move into types of media. If you look at a tube and it's got media in it and it's gelled at an angle like that, it is not a mistake. It's been done that way purposely. This tube is called a slant, and that slant typically has what's called a deep butt. Here's a deep butt, okay? And typically what you do is you take a needle and you inoculate the deep butt, and what that does is it reveals the anaerobic properties of the organism. Then you take a loop and you inoculate the slant, and that reveals the aerobic traits of the organism. So a single tube gives you information about 2 different events that an organism carries out, both anaerobically and aerobically. So that's pretty neat. These are the types of media that we deal with. Let's just get started here. Complex media. If you have a petri dish and we cannot describe to somebody exactly what's in that petri dish, such as you can't say, well, it's got glucose, it's got salt, it has potassium sulfate and potassium disulfate, if you cannot list chemically what's inside a media, then we call that complex. So that means that complex media has a lot of things in there. It has a lot of different amino acids, it has a lot of different vitamins, and usually we use this media to culture what's called fastidious organisms. Fastidious organisms are organisms that have a demanding nutritional requirement. So if you have an organism and it needs to have the very best in order to grow, you use a complex media, okay? And as you can see here, we cannot express chemically what's in there. Why? Because there's a whole bunch of different amino acids, a whole bunch of different vitamins that we cannot list, a whole bunch of different sugars. So if we cannot list it, it's complex. And again, why we use it? We use it in order to support fastidious organisms, organisms that have a complex nutritional requirement. Okay. This is what's called a selective media. So in the selective media, you've done something to the media so it allows certain organisms to grow, but not others. So here what we did is we changed the pH of the media. You can see at 7.3, just about everything grows in it. But if we lower the pH, if we make it acidic, only fungal colonies grow. So we have made the media selective by lowering the pH and allowing it to selectively grow fungi and not everything else, okay? So that's an example of a selective media. This is called a differential media. So this is a blood plate, okay? We have what's called alpha, beta, and gamma hemolysis. So in alpha hemolysis, we're talking about the partial lysis of red blood cells. So you can see that we can kind of see through it, but not very well. And also a green sheen develops on top of it. This is alpha hemolysis. An example of an organism that does this is Streptococcus pneumoniae is an example of an organism. Here's beta hemolysis. In beta hemolysis, we have a complete lysis of red blood cells. That's typically a bad guy. So this would be an example of Streptococcus pyogenes, the Streptococcus pyogenes. And then this is gamma hemolysis, in which there's no breakdown of red blood cells. In that instance, Enterococcus faecalis would be an example of that. Even E. coli would be a good example. So they do not break red blood cells. So these organisms, if they're breaking down the red blood cells, what they're doing is they're breaking in order to steal the heme and steal the iron for the electron transport chain. And the reason that this is a problem is because if it's doing it on a petri dish, chances are it's doing it in your own body as well. Okay, so those are scary little guys. But the point is that this is the example of a differential media. In a differential media, more than one type of organism grows, but you can tell them apart. So that's a differential media. Here's another example of a differential media, okay? In this one, we have sugars, we have a pH indicator, and we have an upside down tube called a Durham tube. So if the pH is red, that means that it's alkaline to neutral. And if it's yellow, that means that it's acidic, okay? So you can see that this organism, when I was put in here, it did not grow well, okay? Or if it grew, it did not catabolize sugars. So there was no fermentation. So the media stayed red, and there's no gas in the Durham tube. But a different organism over here did catabolize the sugar. It acidified the media, it turned it yellow, and it released carbon dioxide into the Durham tube, okay? So this is another example of a differential media. We inoculate with two different organisms, and it was able to differentiate between the two organisms based on whether it could ferment or not ferment the simple sugars inside the media. This is an exciting example because here we have, this is called MacConkey agar. Okay, so notice that here we have E. coli, and over here we have Staphylococcus aureus. Okay, one grows and the other one does not. Now, if you do not remember, E. coli is gram-negative and Staphylococcus aureus is gram-positive. So the dyes in MacConkey are selective for gram-negative organisms. In other words, it only allows gram-negative organisms to grow. Notice the second plate, also MacConkey agar. Here we have E. coli, and over here we have Salmonella enterica. They both grew. So that means that they're both gram-negative. However, notice that the E. coli turned a pink color, and Salmonella enterica acidified the media, turning it yellow. So in this instance, the MacConkey agar is functioning selectively, while over here, it is functioning differentially. So this is a neat media because it does not one thing, but two. It functions selectively and differentially. Over here, we have a nutrient agar plate, and we plated both organisms here, and this is a positive control. This is allowing us to assess if the absence of this Staphylococcus aureus is due to the selectivity of the MacConkey agar or because we made a mistake. If it grows here, but it doesn't grow here, it was because of the selectivity of the MacConkey. However, if it doesn't grow here and it doesn't grow here, that means the aureus is not functioning properly and we are making a mistake. So that positive control allows us to assess the selectiveness of this particular MacConkey agar. Okay. There's a summary on Coulter counters and flow cytometry. I find this very exciting because that's what allows us to do CBCs, complete blood counts. The lasers diffract differentially on the different types of cells. In other words, it identifies them and then it counts them. So you can do a complete blood count, or you can do it in research and use an immunofluorescent tag on the cells and then count the expression of a particular protein in a set of cells. So these Coulter counters and these flow cytometers are a fantastic way of enumerating bacteria. Okay, let's finish here. Okay, this is a spectrometer. This we use what's called Beer's law. And Beer's law in chemistry tells us that the absorbance, how much light is absorbed by a liquid, is proportional to the concentration of a solute in that liquid. Okay. The reason that this is important is because if you can build a standard curve, you can use a spectrometer to calculate the cell density and find out what the number is immediately. So spectrometers are very, very useful in microbiology because they can allow you to calculate the cell density quantitatively based on the turbidity of the liquid. Okay, so that is chapter 6. I hope I didn't bore you too much. Thank you, and have a wonderful day.